Bacterial cell wall polysaccharide, that is bacterial cell wall structure, composition and types. Cell wall is an important structure of bacteria. It gives shape, rigidity and support to the cell. On the basis of cell wall composition, bacteria are classified into two major groups, that is gram positive and gram negative. Next, coming to the types of cell wall. There are two types of cell wall based on the cell wall. One is gram positive cell wall and gram negative cell wall. In gram positive cell wall, the cell wall composition of gram positive bacteria is one is peptidoglycan, lipid and teichoic acid. When coming to the gram negative bacteria, the cell wall composition of gram negative bacteria is peptidoglycan and outer membrane, lipids, protein, lipopolysaccharides. Composition of the cell wall. First one is peptidoglycan. Peptidoglycan is a sporous cross-linked polymer which is responsible for strengthening of cell wall. Peptidoglycan is composed of three components. One is glycan backbone, second one is tetrapeptide chain, that is chain of 4 amino acids linked to N-acetyl monoxide, that is NAM. Third one is peptide cross linkages. Glycan backbone is the repeated unit of N-acetyl muramic acid, that is NAM, and N-acetyl glucosamine, that is NAG, linked by beta glycosidic bond. The glycan backbones are cross-linked by tetrapeptide linkages. The tetrapeptide is only found in NAM. More than 100 peptidoglycan are known, with the diversity focused on the chemistry of peptide cross-linkage and inner bridges. Although the peptidoglycan chemistry vary from organism to organism, the glycan backbone is NAG and NAM is same in all the species of bacteria. The gram-positive bacteria, the arrangement of polysaccharide is and tetrapeptide, that is L-alanine, first position in both gram-positive and gram-negative bacteria. D-glutamic acid, that is second position. Third one is D-aminoamic acid and lysine, that is third position. D-alanine is the fourth position. In gram-negative bacteria, peptide cross-linkages occur between diaminoamic acid, that is third position, of one glycan backbone and D-alanine of adjacent glycan backbone. In gram-positive bacteria, peptide cross-linkage occurs by peptide interbridges. The type of number of amino acids in the interbridges vary among the bacterial species. Next one is teichoic acid. Teichoic acid is water-soluble polymer of glycerol or ribitol phosphate. It is present in gram-positive bacteria. It constitutes about 50% of dry weight of the cell wall. It is the major surface antigen of gram-positive bacteria. It is an additional layer present in gram-negative bacteria. It is composed of lipid bilayer, protein and lipopolysaccharides layer. The functions of outer membrane is structural component of gram-negative bacteria, the gram-negative cell wall. Lipopolysaccharide, that means LPS, is an endotoxin produced by gram-negative bacteria, and lipid A is the antigenic. Next one is lipopolysaccharide, that is LPS. LPS is attached to outer membrane by hydrophobic bond. LPS is synthesized in cytoplasmic membrane and transported to outer membrane. LPS is composed of lipid A and polysaccharide. And lipid A, it is a phosphorylated glucosamine disaccharide, that is polysaccharide. It consists of core polysaccharide and O polysaccharide. The examples for gram-positive bacteria is Micrococcus, Staphylococcus, Streptococcus, etc. The examples for gram-negative bacteria is E. coli, Salmonella, Klebsiella, Shigella and Pseudomonas. Glycoproteins. Proteoglycans or glycoproteins are macromolecules of the cell surface or extracellular matrix, in which one or more glycosaminoglycan chains are joined covalently to a membrane protein or a secreted protein. The glycosaminoglycan moiety commonly forms the greater fractions of the proteoglycan molecule, dominates the structure, and is often the main site of biological activity. In many cases, the biological activity is the provision of multiple binding sites, rich in opportunities for hydrogen bonding and electrostatic interactions with other proteins of the cell surface or extracellular matrix. Proteoglycans are major components of connective tissues, such as cartilage, in which there are many non-covalent interactions with other proteoglycans proteins, and glycosaminoglycans provide strength and resilience. Glycoproteins have one or several oligosaccharides of varying complexity, joined covalently to a protein. They are found on the outer face of the plasma membrane, in the extracellular matrix, and in the blood. Inside cells, they are found in specific organelles such as Golgi complexes, secretory granules, and lysosomes. The oligosaccharide proteins of glycoproteins are less monotonomous than the glycosaminoglycan chains of proteoglycans. They are rich in information, forming highly specific sites for organization and high affinity binding by other proteins. Glycolipids. Glycolipids are membrane lipids in which the hydrophilic H groups or oligosaccharides, which as the glycoproteins act as specific sites for organization by carbohydrate binding proteins. So next, coming to the glycoprotein subheading. The carbohydrates can be attached to proteins to form glycoproteins. A glycoprotein is a type of protein molecule that has had a carbohydrate attached to it. The process either occurs during protein translation or as a post-translational modification in a process called glycosylation. The carbohydrate is an oligosaccharide chain, that is glycan, that is covalently bonded to a polypeptide chains of a protein because of the OH group of sugars. Glycoproteins are more hydrophilic than simple proteins. This means glycoproteins are more attracted to water than ordinary proteins. The hydrophilic nature of the molecule also leads to the characteristics folding of the protein in tertiary structure. The carbohydrate is a short molecule, often branched, and may consist of simple sugars, example glucose, galactose, mannose, xylose. The amino sugars are sugars that have an amino group. They are called as amino sugars, that is such as N-acetyl glucosamine or N-acetyl galactosamine. And acidic sugars, sugars that have the carboxyl group such as sialic acid or N-acetyl neuromic acid. There are two types of glycoproteins. One is O-linked, another one is N-linked glycoproteins. Glycoproteins are categorized according to the attachment site of the carbohydrate to an amino group in the protein. That is O-linked glycoproteins. The O-linked glycoproteins are ones in which the carbohydrate bonds to the oxygen atom of the hydroxyl group, that is OH group, of a R group of either the amino acid threonine or serine. Threonine or serine la idu attached. O-linked carbohydrates may also bound to hydroxylation or hydroxyproline. The process is termed O-glycosylation. That means O-linked glycoproteins are bound to sugar within the Golgi complexes. When coming to the N-linked glycoproteins, the glycoproteins have a carbohydrate bonded to the nitrogen of the amino group, that is NH2 group, of R group of amino acid, asparagine. The R group is usually the amide side chain of asparagine. The bonding process is called N-glycosylation. The N-glycoprotein gains the sugar from the endoplasmic reticulum membrane and then are transported to the Golgi complex for the modification. So difference என்ன அப்படின்னு பார்த்தீங்கன்னா ரெண்டுமே வந்து அமினோ குரூப்ல அட்டாச் கூடிய கார்போஹைட்ரேட். ஸோ அமினோ குரூப்ல OH குரூப் ஆர் கார்பாக்சில் குரூப்ல அட்டாச்னா தேர் கால்ட் அஸ் O linked. அமினோ குரூப்ல அட்டாச் ஆச்சுன்னா அது N linked அப்படின்னு சொல்றோம். Then P-glycosylation occurs when the sugars attach to a phosphorus of phosphoserine. Then C-glycosylation is when the sugar attached to the carbon atom of an amino acid. An example is when sugars mannose bonds to the carbon in tryptophan. Next, Glycation. Glycation is when glycophosphatidylinositol, that is GPI, glycolipid attached to the carbon terminus of polypeptide. Glycoprotein examples and functions. Glycoproteins function in the structure, reproduction, immune system, hormones, and protection of cells and organisms. Glycoproteins are found on the surface of lipid bilayer of cell membranes. Their hydrophilic nature allows them to function in the aqueous environment, where they act in the cell-cell organization and binding of other molecules. Cell surface glycoproteins are also important for cross-linking cells and proteins, example collagen, to add strength and stability to the tissue. Glycoproteins in plant cells are what allow plants to stand upright against the force of gravity. Glycosylated proteins are not just critical for intracellular communication. They also help organ systems communicate with each other. Glycoproteins are found in brain gray matter, where they work together with oxygen and synaptosomes. Hormones may have glycoproteins. Examples include human chorionic gonadotrophic and erythropoietin. Blood clotting depends on the glycoprotein prothrombin, thrombin, and fibrinogen. Then cell markers may be glycoproteins. The Mn blood groups are due to two polymeric forms of the glycoprotein, the glycoprotein A. The two forms differ only by 2 amino acid residues. Yet that is enough to cause problems for persons receiving an organ donated by someone with a different blood group. The major histocompatibility complex MHC and H antigen of the ABO blood group are distinguished by glycosylated proteins. Glycoproteins A is also important because it is the attachment site for Plasmodium falciparum, a human blood parasite. Glycoproteins are important for reproduction because they allow for the binding of sperm cells to the surface of the egg. Mucins are glycoproteins found in mucus. The molecules protect sensitive epithelial surfaces, including the respiratory, urinary, digestive, and reproductive tracts. The immune response relies on glycoproteins. The carbohydrate of antibodies, that is, which are glycoproteins, determines the specific antigen it can bind. B cell and T cells have surface glycoproteins which binds antigens as well. Glycosylation versus glycation. The glycoprotein gets their sugar from an enzymatic process that forms a molecule that would not function otherwise. Another process called glycation covalently bonds sugars to proteins and lipids. Glycation is not an enzymatic process. Often glycation reduce or negates the function of the affected molecule. Glycation naturally occurs during aging and is accelerated in diabetes patients with high glucose level in their blood. Comparison of N-glycosylation and O-glycosylation. N-glycosylation. It is attached to asparagine residue. Linear protein consumes sequence for attachment. That is conserved core saccharide structure. It contains N-acetyl glucosamine linker. Then it biosynthesized by remodeling of a specific attached oligosaccharide and can be relatively large 12 to 20 monosaccharide residues. And it is extracellular, vesicular or cell surface. The coming to O-glycosylation, it is attached to serine or threonine residues. No linear protein consequences or attachment. No conserved core structure. No core saccharide structure. And N-acetyl galactosamine linker are mostly present in the structure. That is biosynthesized by sequential addition of single monosaccharides. It is usually relatively small. It contains 3 to 6 monosaccharide residues. So it is an extracellular or cell surface. Blood group polysaccharide. A type of complicated and living tissue which holds numerous proteins and cell types. Blood is important in our bodies due to its functions: defend, regulate and transport. It is also known as fluid tissue present in blood vessels. Human blood groups depends on the functioning of glycosyl transferases, enzymes that catalyze the formation of glycosidic bond between the structure and function of the human blood. Specific oligosaccharide antigens attach to the proteins and lipids on the surface of erythrocytes. Those attached to proteins have a serine or threonine residues or ceramide lipid intermediate. The most basic oligosaccharide attached is called the O antigen, also referred to as the H antigen. This O antigen is the base oligosaccharide found in all three blood types AB, A and B. The O antigen is often forms that is lipid, glucose, galactose, N-acetylglucosamine, galactose and fucose. The blood type O only has the O antigen attached to the red blood cells. Blood type A is formed through an addition of A antigen which has N-acetylgalactosamine, that is GalNAc, glycosidically bonded to a O antigen. Similarly, for blood type B, the B antigen has an additional galactose forming a glycosidic bond to the O antigen. In both A and B blood types, the new antigen forms an alpha-1,3 linkages to the outermost galactose component of a O antigen through the help of glycosyl transferases. That is, GalNAc transferases added the extra N-acetylgalactosamine for the A antigen, while Gal transferase adds the extra galactose for the B antigen. Genes in the person's DNA code for the specific glycosyl transferases to allow for the addition of antigens A and B to the O antigen. If a person's genes do not allow for a coding of type A or B transferases, then that person will have O type blood. The genes that codes for a GalNAc and galactose transferases are exact. But for the three amino acids, the strong similarity between the two enzymes shows they are related through divergent evolution. Blood types. We can categorize our blood types into four groups: A, B, AB and O. These four groups are considered to be the four phenotypes that one may possess. These four phenotypes can produce 6 different genotypes that each person's can be AA, AO, BB, BO, AB or OO. On the structure or on the surface of red blood cells, there are two types of antigen, that is A and B. Each type has own properties. If a blood cell is type A, the surface of cell contains antigens for type A, and the body will produce antibodies for the type B. And vice versa for type B, type AB contains both antigens on the surface and has no antibody. Blood type O has no antigen and thus have both A and B antibodies in its system. In this regard, blood type O may be the universal donor and blood type B, AB is the universal recipient. We can also look at the property of Rh protein, whether it is absence or presence in our blood. If a blood type has Rh protein, then it is positive. If a blood type does not have Rh protein, then it is negative. The Rh factor is more important if the mother is to have the second child. This is because if the mother is Rh negative and her first child is Rh positive as present of Rh protein, the mother will produce antibodies, specific immunogenic proteins, for the Rh protein. This will not affect the first child since the mother will only produce this antibody after the child is born. However, if the mother is to have the second child with Rh positive trait as well, the antibodies in the mother will cross the placenta and attack the child. This is because the antibodies for the Rh protein are specific proteins called memory cells that are small enough to pass the placenta. This will result in the miscarriage. One way around this is to give a mother Rogam antibody after a birth. This is a synthetic protein antibody that will destroy the leftover fetal cells before it has a chance to interact with the mother's immune system. This inhibits protein antibody production towards the Rh factor in the mother and will allow subsequent Rh plus children to be born from that mother. Universal donor and universal recipients. Blood type O is the universal donors due to its versatility of having no molecules on the red cells, that is red blood cell surface, which will not trigger any immune responses. Therefore, type O blood can be donated to any of the other 4 blood types, earning its name. Blood type AB is the universal recipients due to its lack of antibodies that recognize type A and type B surface molecules. AB can receive blood from any of the other 4 types of, earning its name of universal receiver. AB, however, also tends to be the rarest blood type out of the 4. AB positive is true universal receiver, able to receive all types of blood regardless of type Rh antigen. However, O negative is only compatible with itself but able to donate to everyone. Red blood cells versus blood plasma. Like red blood cells, compatibility recipients can receive blood plasma from the same blood type. However, unlike with red blood cells, the plasma has a converse compatibility. Blood type O can receive plasma from every other blood type, while blood type AB can donate blood plasma to any blood type. The structure of blood group polysaccharide, that is O blood group, it contains galactose, fucose, N-acetylglucosamine, galactose, glucose and lipid. In the blood type A, galnac, galactose, fucose, N-acetylglucosamine, galactose, glucose and lipid. In blood type B, galactose, fucose, N-acetylglucosamine, galactose, glucose and lipid. Binding site.